My name is Giovanna Scapin. I am the chief scientist at the website of a company that's called Nanoimaging Services. And what we do in this company is basically try to enable every process of drug discovery through cryoEM. We help researchers of pharmaceutical biotechnology companies to move into cryoEM. And we try to bring the power of cryoEM to every step on the development of new drugs. And we try to do it in a very informative way with our clients. We look at every steps, and we look at many different aspects of drug discovery, from antibody development all the way to high structure solution and refinement. I want to give a little bit of introduction of cryoEM drug discovery, because this has been my bread and butter for the past maybe 5 years or so. As Sarah mentioned at the beginning, my background is as a structural biologist, and I have been a structural biologist for all my life. And my passion in life is to actually apply structural biology, any kind of structural biology, to the development of new drugs. And I was lucky enough to be able to do it for many years, working for a big pharmaceutical company, and lucky to continue doing it now while I work for Nanoimaging Services. So the development of a new drug takes many years, so we know that, and many, many different steps. And I summarized them very shortly and briefly in this arrow up there. And I talked very often of the contribution that structural biology can give to a drug discovery effort. And most of these contributions are at the very beginning of the drug discovery, when we talk about target identification, hit identification, and lead optimization. Now, in the past, I think, few years, we have seen many, many examples that prove how much cryoEM has contributed to the early stages of drug discovery, and how much of the conformational and size space has cryoEM brought to life, giving us access to structures that a few years ago were completely unthinkable, not just of membrane proteins or larger assemblies, but also things in motion, molecular machines, things that move in order to carry out their own functions, and we are now able to see them. So the role of cryoEM at the very early stages of drug discovery, so structure, target identification, when we want actually understand the biology of the system, and understanding initial binding modes, is clearly established. There is really not too much that we can say or do about it. What we want to do now is to move in this third step, which is the lead optimization, and it's more of a medicinal chemistry realm at the moment, right? So we want to be able to provide structural support using cryoEM for a real structure-based drug design. And what is required in this step of the drug development is the capability of providing structures at high resolution with a very high throughput and with a good timing. The resolution I'm not even going to talk about, because we know how important it is. If we want to see atoms and ligands and bonds, we need to be at high resolutions, but we are moving there with cryoEM more and more often. I want to just make a little comment about timing and throughput that basically ties back to the fact that cryoSPARC Live provides a big support in this development. Why do we need to be fast? And why do we need to be reliable? And also, most importantly, we need to be very early and very proactive in the process. Because if we want to include the structural and modeling information into this cycle that I identify on the left, which is basically a drug development cycle in which we form some hypotheses about a ligand, we design these new molecules, chemistry synthesizes the molecules, and then biology assays and defines the activity of all these molecules. We need to be really fast, because chemistry and biology move really fast when we are in the range of drug discovery. And so if you want to be part of this cycle, we need basically to keep up with the speed in which these molecules are designed, tested, synthesized, tested, and then we want to provide real-time information and inform the further hypothesis and further design. Crystallography is really fast. It's still much, much, much faster than cryoEM. If you have a system in place, you can get several structures a week. CryoEM is still quite slow. It's not quite there yet. Even if you have a system in place, it's so much dependent on sample preparation, grid preparation, and a lot of other factors that we can still get maybe a structure a week, maybe a couple of structures a week, but it's definitely not sufficient to be consistently active in the structure-based drug design cycle. Why is not like that? Why it's still slow? So as I mentioned, there are different things, right? So there is a whole upstream process of sample preparation, optimization of the sample quality, grid preparation, optimization of the grid, and everything else that it's widely discussed in many other forums, and I will not touch it upon here. But one thing that I'm going to touch upon here is the accelerating the process of data collection, data interpretation, and generating a map. So this is actually a pretty old slide that I had done, maybe pretty old, 3 years ago, in which I was asking for automation in the processing world, because human interventions, as I pointed out, was still very much required for particle picking, selecting the 2D classes, go back and recalculate the 2D classes, selecting the particles again, evaluation of the heterogeneity, evaluation of the 3D model and everything else. The pre-processing part, so all the way up to CTF calculation, was actually somewhat automated. There are a couple of systems that do that altogether, but then when we got to the point of particle picking, we are stuck of doing it by hand. And since you will do it once and be done, you will have to wait until the end of the data collection, which could be 2 days, 3 days. And so everything got slowed down a lot. That's why cryoSPARC Live really, to us in drug discovery, made a big impact, because now we have everything automated, right? And we can basically go from data collection to map calculation very fast, and most importantly, hands-off, which means that we can start the data collection on the microscope, we start the cryoSPARC, and then we go on and do other things, and without having to worry about keeping on touching and generating different images and different 2D classes. And I just want to give you a very brief example of how we use cryoSPARC Live in my company. Our mantra is a cryoSPARC Live for every autoloader microscope that we have. And at this point, we have 4, maybe soon to be 5. And so we have cryoSPARC Live running every time we do a data collection on one of these microscopes. We have 2 classes of microscopes, glaciers and the cryos. And cryoSPARC is widely used in all the scopes with slightly different flavors. So for the glaciers, which is what I mostly use in our facility here in Woburn, we do a lot of negative staining, interestingly enough. And we use cryoSPARC Live to provide a real-time analysis of the samples, so from grid preparation all the way to 2D classification, in about 3 hours. And so we can go through many different samples in a day, and mostly we can provide our clients with real information about the status, the quality, the type of the samples that are not easily extractable just by the micrographs. We do a lot of preliminary work for vitrification. We see a lot of very difficult samples that are seeing cryoEM for the first time. And so there is a lot of screening that we need to do for optimal condition. And using the cryoSPARC Live, we can, again, running in parallel with an overnight data collection. And by the end of the data collection, understanding really the sample quality, if there is an issue of preferred orientation. And we can, again, provide the client with a much better characterization of the grid preparation, of the quality of the grids, or the vitrification system that we have utilized to make those specific grids. At the In San Diego, where we have our cryos farm, we use cryoSPARC Live for, again, different goals. If we have a very good sample that we know it's good, we do what was basically shown in the movie. We started in parallel with the data collection, and we get to a full 3D map in 24 to 48 hours, which is not several structures a week, but we are getting there, because if every sample behaves well, we can actually do that. The other good thing, as we mentioned before, is that we can stop the data collection if we realize that the ice is too thick, the classes are not good. All information that we can't get just by looking at the micrograph as they came across. And that saves a lot of time, because we can either switch to another grade or just stop the data collection and save the client money, basically. For samples that are suboptimal, we use cryoSPARC to evaluation of the sample as the data comes in again. And as before, we can stop the data collection or change it to other grades or propose other solution to the client, or internally decide how to move. And with that, I hope I gave you a good overview of what we do and why we use cryoSPARC Live. And I'll get back to, I think, Ali. Yeah.